Hey guys, this is Kendall Terry. This is DNA replication. So we start by looking at the simplest form of DNA replication, which would be the prokaryotic cell. Now we know that prokaryotic DNA is circular. Now don't get confused here, it is still a double helix, just like Watson and Crick discovered and all that. But for simplicity's sake, I just drew it as two circles showing the different strands. There is an origin point on the DNA and a termination point. And the origin is the initiator protein rich in AT nucleotides. That's important because A and T come together in just 2 hydrogen bonds as opposed to C and G that come together in 3 hydrogen bonds. So it's a little bit easier to separate the AT nucleotides. So then you get the binding of 2 replisomes at the origin point, and it will actually start at one point and go in opposite directions, as you can see in the picture, and it will actually do replication both ways. So it starts at that point, it's obviously producing strands of DNA as it goes until eventually it gets to that termination point, and it will separate then, and you will have 2 DNA strands. They are identical to each other, and they were formed at the same time. It doesn't start and then go all the way around. It actually starts and then stops over here so that it makes 2 by doing kind of half circles. Okay? This is actually what it looks like. That replosome, which is this complex right here, is quite intense. One thing that we know is that there's a DNA gyrase over here. Now that purpose is to keep the DNA strand from supercoiling, because as this is opened up, it's kind of tightening that helix, and if there wasn't something out here to stabilize it, the DNA would just start to wind around each other. The best description of that, which you may not know, is a phone cord. If you've ever used a phone that actually has a cord to it, which many of you may not at this point, as that phone continues to get twisted as you answer it and hang up and talk and turn in circles or whatever, that cord will start to wind around itself and get all mangled up. So DNA gyrase's job is to actually keep that from happening, and it'll hold it and stabilize it so it'll kind of supercoil a little bit right here, but it won't let the rest of the strand bind all up on itself. Then you've got DNA helicase. DNA helicase's job is to break those hydrogen bonds and separate that DNA. You can see that here. And then there are some proteins that will stabilize those open nucleotides until DNA polymerase binds the new strand. So you've got this complex here that's kind of holding all this together. On this strand up here is the simplest one, and this is called the leading strand. It's the 3' original strand, and it's making a 5' strand. DNA polymerase 3 is bound to it, and it is just binding away at DNA bases. DNA polymerase binds in a 5' to 3' direction, meaning it has to start at the 5' end and it has to go to the 3' end, which is fine up here. That's not a problem. Down here, though, that is a problem, because now your original strand is the 5' strand. So DNA polymerase, to just go in that same direction, would have to be able to bind in a 3' to 5' direction. It can't do that. So what it does, down here, you'll see this, is it does it kind of in chunks, okay? So it will bind and make a little bit, and then it'll have to stop, and it'll bind and make a little bit and stop, and bind and make a little bit to make it in the 5' to 3' direction, which is what it does up here. So you still got DNA polymerase 3 that will make it, but it has to have a starting point. That actually is put together right here at primase, and it puts a primer. That is not going to be what's left at the end, but it's what gives it DNA polymerase the ability to start. So the primer, and then it pushes a little bit, and then it eventually will stop, and it'll have to get another section and do the same thing again. So you end up then with a chunk of DNA that has a primer attached to DNA. And then there'll be a gap. So that gap has to be filled in, and the primer has to be taken off, and DNA has to be put on. So DNA polymerase 1 will come by, remove that primer, and fill in the DNA bases that should be there once the primer is removed. The primer is an RNA primer. So RNA can't be there. So DNA polymerase removes the primer, adds the bases that should be there. There's still going to be a gap right here. Actually, on this one, it would be right here, not right there. So finally, ligase comes in and seals up that gap and creates that phosphodiester bond between the sugar-phosphate backbone there. So you've got a gap here that's going to be left once this is all done and that primer's filled in. And ligase will finally seal that final strand. This is called the lagging strand down here because it has to make these pieces that then have to be fixed with the primer removed, and then finally ligase has to seal it up. So that will give you then two strands, okay, that were both made in this process. And this is how DNA is replicated, or a copy. Now it does it simply by adding the base pair that's open. So you've got a strand that has the code. The new strand will be attached to it based on that code. If there's a T, an A will bind. If there's an A T will bind. If there's a C, a G will bind. And if there's a G, a C will bind. And it will fill in and make an old strand connected to a new strand. Up here, old strand connected to a new strand. I encourage you to go back and look at that and make sure you get it. In eukaryotic replication, the sheer amount of DNA requires multiple origins. So now instead of just that one circular piece of DNA with one origin and one termination, now you've got this huge DNA strand, and it's not a circle. It's just one strand with endpoints. Okay? So, and it's massive. So for your cell to be able to replicate its DNA, it can't just start at one point and replicate along. It'll actually start at multiple points and be replicating all over it, and then eventually it'll seal up all the strands with ligase. Despite additional complexity, because of the size, because of the multiple origin points, the action of the replosome seems to be similar to that described in E. coli, which is what we were looking at earlier. The replication fork is also essentially the same. That's that point that helicase separates the DNA. That's called the replication point. That's essentially the same as well. The end of the DNA is called the telomere. The telomere, which would continually shorten due to the primer on the lagging strand. So remember, you've got this primer that had to be attached to the lagging strand to make DNA. So when that primer gets removed, there's not that gap can't be filled in by DNA polymerase because there's an end there. It can't fill in. Okay? So if under this circumstance, without some help of something, and I'm going to show you what that something is, what would happen over time is your DNA would continue to shorten. And if your DNA continued to shorten, eventually you would lose important genetic information, and that would cause various problems later on in life. In fact, there's some diseases that are being shown due to shortening of the telomere end. So how does the cell deal with this? Well, it deals with this through DNA telomerase. Telomerase is right here, and it's this protein. So here you've got the DNA, and you've got this lagging strand that is missing. So if it continued to replicate from here, the next strand would be shorter, and so on and so forth until you'd end up with some problems. So how does the cell deal with that? Well, this enzyme here, this green enzyme, is called telomerase. And telomerase has a built-in RNA template. So it will bind, you can see the TTG, so it has an AAC that binds, okay? And then it has an additional code, CCAAC, and that can be longer if it needs to be. But it will bring in then bases that will grow the end of this strand. As it grows the end of that strand, it will move down and it will leave an open point now. So now you're ready to synthesize the next repeat of DNA. So you actually grow the end, the telomere end will actually be grown so that it's longer, so that as eventually there's still going to be a shorter strand, but that shorter strand is going to be these repeats of T's and G's, and it's not going to be something important. So although there's still going to be a difference down here at the end, it will take care of it not damaging the DNA down here. This enzyme is called telomerase. Now, what they have also shown is that telomerase activity, okay, gets messed up. When telomerase activity gets messed up, either it's doing uncontrolled growth of the DNA, which would give a bunch of nonsense DNA, or it shuts down altogether to where it's no longer present, there are problems that develop, one of which would be cancer. Whoa. So let's go ahead and talk about then some problems. So if you have DNA that's been mutated, there has to be some repair. So a mutagen is any agent that increases the number of mutations above background level. Background level is just mutations that occurred. Polymerase has a pretty good proofreading ability and can get it right. Occasionally it'll get one wrong. But above that level, there's things that will cause it to get it wrong. One of which is UV light. So in UV light can cause two T's that are next to each other to bind to each other and crank the DNA, and that's called a thymine dimer. The cell has an enzyme that battles this. Photolase is an enzyme that uses visible sunlight energy to break that dimer and reset the DNA the way it's supposed to be. So that's one way that your cell will repair that DNA, because as you go outside and you're in the sunlight, you're going to have some of these thymine dimers that develop, but there's an enzyme in your cells that's going to cause that thymine dimer to be broken and reset the way it's supposed to be. If left like this, it would cause problems, and if enough of these happen, or this enzyme starts to be mutated where it doesn't work, then you can lead to stuff like cancer. That's one of the causes of cancer. Finally, we're just going to do an overview of repair. So if you have something that's messed up, you've got some DNA bases that are inserted incorrectly, occasionally that happens, there's a system of checking it. So the first is a recognition of damage. The protein that does this is called a UVR-ABC complex, and this complex will kind of read the DNA. Then if it notices a damage, there'll be a removal of that damaged region by DNA polymerase 1 or 2, either one. And then you'll have a resynthesis using the information of the undamaged strand with DNA polymerase 1 or 2 as well. So you've got DNA polymerase 1 and 2 that really go through and proofread that strand, make sure there's no damages. The UVR-ABC complex will give a flag of, hey, there's a damage here. DNA polymerase 1 or 2 will come along, take that strand out. Polymerase has the ability to break the phosphodiester backbone bonds and remove that piece that's messed up, and then insert and reconnect up those phosphodiester bonds, that DNA strand, thus making it correct again.